Okay guys, in this next section of the video, I'm going to tell you about the gram-positive and gram-negative cell wall. So starting with gram-positive, remember gram-positive bacteria will stain purple with the gram stain. Some examples of pathogens that are gram-positive are Staphylococcus aureus, which we all do carry this on our normal flora, but also there are pathogenic strains of this, like you guys might have heard of MRSA, which is a methicillin-resistant Staph aureus. Bacillus anthracis, this causes anthrax. Clostridium difficile, some of you who work in the medical field, you might call this C. diff. And Streptococcus mutans, which actually causes dental cavities. So here is what the gram-positive cell wall looks like. I'm going to do a drawing here. So first of all, we have our regular cell membrane. This is our plasma membrane. And remember, this is a phospholipid bilayer. And facing the inside here, we have the lipid tails. And on the outside here, these little round things, these are the phosphate heads. And just to remind you, the purpose of this cell membrane, the phospholipid bilayer, is to serve as a barrier to block things from freely entering or exiting the cell. So it is permeable only to specific things, semi-permeable is what we would call it, okay, allowing only certain things to pass in and out. Next, outside of this phospholipid bilayer, so here's the inside of the cell, just to be clear. Inside, and up here, this will be the outside. Okay, so next, we have a very, very thin space. Okay, here's a thin little space. This is actually a gel-like material, which we call the periplasm. In gram-positive bacteria, just to remind you, this is gram-positive, this space is very thin, okay, not thick at all, very thin, and there may be a few proteins inside of it, just very few, okay, so maybe some proteins inside of there. Now, on the outside of the very outside of the cell wall, here's where we have that peptidoglycan that we did in the last video. Okay, so we have many, many, many layers of it in gram-positive. All right, and remember, they're all linked together. All right, so I'm not going to draw the whole thing. You get the picture. So this is a thick peptidoglycan. When I say thick, I mean this is about 30 to 40 layers of those glycan chains, very thick. The last thing that we have in gram-positive bacteria are these chains here that I'm going to put inside. These are called teichoic acids. And there's also a second type. These are called the lipoteichoic acids. All right, and the teichoic acids and the lipoteichoic acids, they're going to help stabilize this really thick peptidoglycan. Okay, so overall, pretty picture per usual. We have, again, the inside, the cell membrane is here, followed by a very thin periplasm, And remember, this holds a few proteins inside of it. And then here we have this very thick peptidoglycan. And these little red rope-like things are the teichoic acids and the lipoteichoic acids, which are unique to gram-positive bacteria. And once again, the best way to learn this is by drawing it. It helps you to memorize it, okay? All right, now let's talk about the gram-negative cell wall. So some examples of gram-negative bacteria: E. coli, Salmonella typhi. You guys have heard of Salmonella, I'm sure, from eating raw chicken. Neisseria gonorrhea. This causes a sexually transmitted disease. And Pseudomonas aeruginosa. This is a common hospital-acquired infection. It usually only affects immunocompromised individuals. Now the gram-negative is a little bit more complicated. So again, here we have the cell membrane, the phospholipid bilayer. So we're talking about gram-negative now. Okay, so phospholipid bilayer. Now, again, this is the inside of the cell. After the phospholipid bilayer, we're going to have a periplasm. But this periplasm is very thick. So a thick periplasm. And again, the periplasm is a gel-like material. And there's a lot of really important little proteins embedded within it. And these proteins have a lot of important functions. Now, the next thing we're going to have within the periplasm, inside of it, that's where we're going to find the peptidoglycan. Okay, so the peptidoglycan, P-glycan, is inside the periplasm, but it's very thin. Okay, maybe like 2 to 3 layers of it. Then the last thing, this is so different, okay? Gram-negative bacteria actually have a second membrane. An outer membrane is what we're going to call it. So really, this one down here, this was an inner membrane, and this one's the outer membrane. But what's unique about the outer membrane? The first layer, again, it's a bilayer, but the first layer is going to be phospholipid. Phospholipid. The second layer is this special material. Okay, I'm going to draw, it looks like a squiggly line right now, called a lipopolysaccharide. And I'm going to abbreviate this LPS. So the LPS is in the outside of the outer membrane. So remember, lipo, this is the lipid. So the lipid part faces the inside. And the polysaccharide, this is a hydrophilic sugar, and that faces the outside. So LPS is unique to gram-negative bacteria. And we're going to talk about this a lot in other lectures, so make sure you remember LPS is unique to gram-negative bacteria, and it's in the outer membrane. Okay, so again, pretty picture here. We have the inner membrane down here. It's a regular phospholipid bilayer. Next, this big purple chunk here, that's the periplasm. And within the periplasm right here, that's where you'll find a thin peptidoglycan. Then on the outside, okay, we have the outer membrane, outer membrane. The first layer here are those phospholipids. And then this stuff on the outside here, remember that's LPS, lipopolysaccharide. Okay, so I hope this video was helpful in helping you to memorize and learn the different structures in the gram-negative and gram-positive cell wall.